Good. So hello, everyone. It's very hard to say good morning, good afternoon, and good evening for a lot of people. And myself, Sabir Gan, I'm actually chair of Chemical Application Group. It's a pleasure to introduce my two speakers today for this meeting. Both speakers are going to talk about a very important class of materials. They're biomedical, they're mostly used in biomedical application. And the common, I would say, principle behind these materials are that they are based on fibers. And we will hear from both speakers how they address development of these fibers by having liquid coat technology and surface modification. And mostly these are platform technologies for localized therapy, diagnostic, and regenerative medicine. So I am really excited to hear from both of them. So before we start the presentation from the speaker, just general rules. Please keep your mics muted. And if you have questions, please type in the name of the speaker and also write down the questions you have for the speaker. And I'm going to read the questions at the end of each presentation, and then we will do Q&A. So the way we are going to organize this meeting, we will have the first speaker, Dr. Edith Perret. She will speak first, and then we will have the QA related to her presentation, followed by Professor Liu's presentation later. So let's start without any delay. We have the first speaker, Dr. Perret. She is actually, I can safely say that she's actually a group leader now at Empa. She started working as a scientist at Empa. I know her very well. She's my colleague, and she's an expert in polymer physics, has a lot of experience in X-ray analytics and development of new polymeric fibrous materials. And she is going to speak today on the topic of very innovative fibers, which are liquid filled, and how they can be used for biomedical applications. So, Dr. Perret, the floor is yours. Go ahead.
Thank you very much for the kind introduction, Sabir Gan. So it's a great pleasure to be here today and present my work to all of you. So here it's very foggy, so I'm actually happy to be inside because outside the weather is not so great. I hope it's better in the States or wherever you are now. So yeah, today I have the pleasure to talk about the revolution at the core, liquid-filled fibers for smart applications. So I'm actually from Empa, like also the moderator Sabir Gan. We are based in St. Gallen. Actually, Empa has 3 sites. So one is in Thun, one in Dübendorf, and one in St. Gallen here in Switzerland. So this is the Swiss Federal Laboratories for Material Science and Technology. We have more than 1,000 employees, a lot of scientists, and of course also several departments. And I'm located in the Materials Midlife department. We have several laboratories that belong to these departments, and both of us, moderator as myself, we are in the Laboratory of Advanced Fibers, and I'm also additionally part of the Center for X-ray Analytics because I have a lot of expertise in that field. I did a lot of synchrotron analytics and also neutron scattering and so on in the past, but not so much recently. More lab-based techniques are now in my focus. So I'm in the Polymer Function and Processing Group, and starting next year, I will also become the interim group leader of this group. So the core expertise of our Polymer Processing Group lies actually in melt spinning. So often industrial partners, they approach us with a specific problem about their products. So depending on the applications, different mechanical properties are needed, and sometimes they also have to be improved for specific products. So it is very important to know how we can tailor the mechanical or chemical properties of a fiber. So we can actually achieve that by treating the polymer or by adapting production parameters or by treating the already produced fiber by, for example, annealing. So all these treatments change the structure of a fiber, and with that then also the properties of the fiber. So this is our core expertise. So we modify applications or like products for specific applications by changing these things. Either we change a polymer or we really focus on the production parameters of the melt spun fibers. But today, I would like to focus on a special type of fiber, which is a liquid core fiber, which is also based on the melt spinning technique. So I will explain to you how we make liquid core fibers out of liquid and polymer pellets with this melt spinning technique, and how they can be used in the end for functional textiles in the medical field as well as for other types of applications. But first of all, let me start asking the question, why would we actually like to have a liquid core? So originally, the idea was actually inspired by this bio-inspired strategy. When you look at, for example, a ladybug, so the insect wings, they are really providing an exemplary bio-inspired strategy for designing microsystems by utilizing fine and flexible liquid core tubular structures. So, for example, these beetles, they have specialized veins that enable this unfolding of the wings. So they just pressurize the veins when they do that. So we can use these examples to think about some pressurized devices that can also unfold some fine structures, for example, in micro-robotics systems. So a possible application of liquid core filaments is in soft robotics. So where, for example, pressure-resistant filaments could act as microhydraulic systems within a restricted space. They could be actuated by a powerful external pump, for example. And in consequence, these liquid core fibers that are embedded into these flexible structures could deform in a predefined way when the fluid is pressurized. So what I show you here are some examples that you can find on the internet of soft robotics devices. So for example, here on the right, you see how a beaker is held up and it can also rotate, for example, or here we have a twist actuator or kind of a spider movement. So there are a lot of different types of movements that would be possible. But how can we actually produce such liquid core fibers? So this has originally been invented actually here at Empa. So it is all based on the original melt spinning technique. So for those who are not so familiar with melt spinning, I quickly explain how this works. So you feed in some polymer pellets in a screw extruder that is heated to a certain temperature above the melting temperature of this specific polymer, of course. So, and then you press it through a melt pump, and then the molten polymer is pressed into the spin pack. And here in the spin pack, this is actually the special design where here we are actually injecting then the liquid directly into the polymer melt. So this was developed here at Empa. And you see a close-up of this specialized spin pack here on the left-hand side. So in yellow is the polymer melt, and in red you see the capillary die. So you can see that actually the liquid meets the polymer melt before it exits the die. So, and you form kind of a core sheath structure. And when it exits the die, you have, of course, the die swell, but then you have the really nice core sheath configuration if everything works as it's supposed to. And then it can cool down this molten strand in a water bath very rapidly. And you draw then the fiber over these so-called goddets, and you wind the fiber up in the end. So what you obtain with melt spinning is really bobbins that contain monofilaments that are really kilometers long, right? So you obtain this just within a very limited time frame. So very small times are enough to obtain kilometers of fibers. This is actually the most used spinning technology in the industry. When you think about like textiles, like woven textiles or knitted textiles, they're all based on melt spinning. But what is new is this liquid core. So this has been developed in 2016. And now recently, this was also upscaled in the industry by an industrial partner from us. They are called Monoswiss, and they are located in Emmenbrücke in Switzerland. And we have several projects together always with Monoswiss, where we also try to bring some liquid core filaments to the market. So some of you might now ask yourself, why should we not just fill a hollow fiber with a liquid? So the reason is shown here on this slide. So it is actually quite slow to fill a hollow fiber with a liquid. So this is actually, you can nicely calculate this with the Hagen-Poiseuille law, how long this takes. And here you see 200 fibers that were actually filled with a red fluid. And you see how slow it is. I mean, imagine if you have now kilometers of fibers that you want to fill, this would take forever. Huh? When you calculate this, you can calculate, for example, for a 10-meter long hollow fiber with a core of 30 microns, then that would take over 3 months with the post-filling when you fill it with the oil, for example. Whereas when you do that with our online technique, you obtain this within a few seconds. So this is also a dimension that is quite typical, for example, for a melt spun fiber, that you have a diameter of about, let's say, 60 microns up to 200 microns, just that you know what we're talking about. So we're talking about quite thick fibers that are applied for different types of functional textiles in the end. So these fibers are also quite strong, and that's why they are also used for these woven fabrics or knitted fabrics. So we had a lot of projects about liquid core fibers in the past here at Empa. We have been working on soft robotics. Then we have also developed some optical fibers where we have also played with different types of liquid cores and also polymer sheath. Then in the medical field, I will also show you today what I've been doing in the last years and where the journey goes now with liquid core fibers for the future. We have also developed some flame retardant fibers that contain some flame retardant liquid in the core, or some dilatant, shear thickening fluids have been developed also to be incorporated into such liquid core fibers. And we are also currently having a project on insecticidal bed nets. I would also like to show you this project quickly towards the end of this talk, so that you also see something else than just the medical applications. Then in the past, we've also been working on some odorant liquid core fibers, so you can also include some perfume agents, for example, inside a liquid core. We have produced, for example, a liquid core fiber with a peppermint oil, which still smells as peppermint a few years after the fiber has been produced. So that works also pretty well. But now let me start and talk about the first project that I'm pushing a lot now, and this is to use liquid core fibers for medical applications. So what are the potential applications of such drug-loaded liquid core fibers? So this slide shows you a bit my ideas where these fibers could potentially find an application in the medical field. Doesn't mean that they will, but I think it really shows nicely the potential of such drug-loaded liquid core melt spun fibers. So that you know what we're talking about, so we have a polymer sheath here, and inside we have a liquid which is filled with drugs. And these drugs, they can diffuse through the polymer sheath into the surrounding if the drugs are quite small. If they're rather big in size, then they can only go somewhere through the open ends of the fiber. So I think they could find applications in transdermal textiles. So you can make some woven or knitted textiles out of them and put them on your skin for using them as drug releasing textiles. Then also sutures might be a good application because suture materials are already made out of melt spun fibers. So it would basically be a replacement, but with an added functionality that you can maybe add an antimicrobial or anti-inflammatory function to the suture itself. I also think that these fibers might be able to be used for pumping devices when you want to locally pump a medication, for example, to a tumor site where you only want some local release. Then recently I've obtained some funding in order to work on hernia repair meshes. So this project will run for 4 years, and we are trying to make hernia repair meshes that are anti-inflammatory as well as antimicrobial based on this new technology. And maybe also from other implanted textiles, this might be something that we can use it for. So this is all still just thinking. We don't know whether they will really find an application in these fields. So why would we like to have actually a drug-loaded liquid core? So when you look at the normal melt spun fibers, there is not such a lot literature actually around how you can actually put drugs inside the melt spun fibers. There is not much done or reported in the literature, but what is standard is that you can try to mix, for example, the drug with the polymer during the extrusion, and then you extrude just the drug-loaded filament basically in one go. But here you have some problems. First of all, you don't have a good control over the release kinetics. There is often like a burst release, so as soon as you implant the fiber, a lot of drugs go out as soon as they come in contact with tissue. And you also have a limited drug reservoir. You cannot fill as much drug as you want inside such a filament. And it's also difficult to load hydrophobic polymers with drugs. And furthermore, the drugs often don't withstand the high extrusion temperatures of during the melt spinning. Yeah, so this is like standard. Yeah. So what people also do sometimes is coating the filament with an additional drug. Yeah. So the advantage of our technique is that you would have in the liquid core a lot of drugs dissolved, and you have like a diffusion controlled release. So if you have good control over how the drug is diffusing through the polymer sheath into the surrounding media, that allows you then to really get a better control over the drug release kinetics over time. You have also a larger drug reservoir. So hopefully we can achieve also a longer drug release than in the current applied monofilaments. It might also be possible to obtain like a responsive release depending on how we actually link the drugs to the liquid core. There might be a possibility to do a covalent bonding, for example, to the liquid, which is only broken when a certain pH is achieved. Ideas like this that we are pursuing at the moment. And advantage is that it is also still weavable and upscalable, this technology. So what have we done so far? I started with trying out different types of carrier liquids. So one was the glycerol, which is quite a small molecule. This came out right away through the polymer, actually. So we have been able to produce such melt spun liquid core fibers with polycaprolactone as the polymer. On the outside as the sheath material, which has quite a low melting point. But the problem is this glycerol comes out through the polymer sheath because it's such a small molecule. So I moved on and used then polyethylene glycol, low molecular weight PEG for the carrier liquid, and that worked pretty well. So we can mix drugs inside that. We started with fluorescein sodium salt, and this was just because this can nicely be seen with different spectroscopy techniques. And then there's also possibility to offline exchange a liquid when you have something that is quite temperature sensitive, like for example a protein or another molecule. You can just attach the liquid core fiber to a pump and then pump the liquid through. But also here we have to say that this is quite a slow exchange technique. It's better if we can produce the fiber in one go directly and put directly the drugs inside here during the melt spinning. Yeah? So here is the typical dimension of such fibers that we have produced. So they were around 180 microns with a core diameter of about 60 microns. Then we do typically a lot of analytics. So as you might have guessed from the introduction that I'm part of the X-ray centers, that I have a lot of expertise in characterization techniques. So we do a lot of X-ray analytics and Raman spectroscopy to understand better the polymer structure, and which affects a lot also the diffusion kinetics of these drugs. Then we analyze the mechanical performance of these fibers because they are, of course, very important for applications in the medical field or also in other types of applications. We do diffusion trials. So we started off with very simple trials where we just cut the fibers, for example, and then study the release over time. And in the future, we'll also test more the in vitro and ex vivo functionalities. So if drugs that we put inside are still anti-inflammatory, for example, or antimicrobial after the melt spinning technique, which is very important. So here I show you just some simple diffusion trials that we did in the beginning. So we have put loops of fibers inside the phosphate buffered saline solution. This was done just to test whether the drugs come out through the polymer sheath into the PBS solution. We immerse these fibers for 24 hours at room temperature as well as at 37 degrees. And then we also cut 20 times 1 centimeter pieces and immerse those in PBS. And then we studied the release after 24 hours. So what we saw is that the small drug molecules like fluorescein or ibuprofen, they come out through the polymer sheath. And of course also more when the sample is heated. But the bigger molecules, they hardly diffuse through the polymer sheath, which has been quite an interesting observation. So also what we've seen is that the structure of the polymer influences the diffusion. So depending on how many amorphous parts you have in that polymer structure or crystalline parts, that affects a lot the diffusion rate, and also the orientation of crystals within the polymer sheath affects the diffusion. So here again, to summarize what we saw, it's very simple, just small molecules diffused through the polymer sheath, and the larger molecules, they can be administered through the open ends of fibers. So one can also then cut these melt spun fibers into small parts, for example, and embed those at a specific location, for example, as local drug delivery applications. That would be also a possibility. So when you look at the standard drug loading techniques, I've mentioned this technique where you just mix the drugs inside a polymer matrix. But this does really only work for very few medications. What is more often done is like a coating. or a grafting of drugs onto the surface. And most of these techniques have the problem that you have a very fast release in the beginning, and then very rapidly it slows down. So often you only get a release within the first few hours, and then everything is gone. So, but for many medical applications, one would actually like to have a more linear targeted release over a long time frame. And we are working on this. So we are doing at the moment this time-dependent drug release, but I cannot share the results because it has not been published yet. And with our liquid core technology, it's safe to say that we can really affect this time-dependent drug release with different techniques. But also there, I can unfortunately not share too much because it's all unpublished and the work is ongoing. But with that, I would like to move on to the second project, which is now away from this medical field. And this is about insecticidal bed nets. But also here it is about releasing an agent into the surrounding. Yeah. So what is this about? So this project has been going on also for 2.5 years now. So it is about controlling malaria. The problem is that a lot of mosquitoes are actually getting resistant against these toxins that are currently used in bed nets. And so what occurs at the moment is that malaria is again increasing worldwide. And this is really a big problem. So we are working on that. So our goal is to actually use also core sheet filaments with a reservoir of active ingredients, either in a liquid core or in a bicomponent fiber in the core, which would then ideally release a toxin in the beginning and then over a longer time until it reaches the end of life. So that's the goal of this project. So what we do, we produce bicomponent filaments as well as liquid core fibers, where we can actually incorporate new types of toxins also inside these liquid core fibers. Also here I cannot share too much, but this is basically the rough idea. So we do a lot of analytics here at Empa, where we study also the diffusion of these toxins, and then we send the fibers to Vietnam, where they're made into knitted fabrics, and then in the States they're tested for bioefficacy, or also in Africa. So it's a lot of samples go around the world for this specific project, but it's very exciting to work on this one. So with that, I'm at the end of my presentation. I've shown you that there are a lot of different types of applications possible for liquid core fibers. I showed you how we can produce such liquid core fibers with the melt spinning technology, and how they can maybe be applied also in the medical field. As a short outlook, as I mentioned, we obtained funding for this hernia repair project, which started. So we are producing novel functional liquid core fibers for this application, and maybe also for other projects to come. So a lot of diffusion trials, structural analytics are involved there, and we will also do more in vitro and in vivo studies. Hopefully all to be published in the coming years. And yeah, with that, I would like to thank the Laboratory 402 for all the hard work they are doing, and also some people from other laboratories at Empa, which helped contributing to this project and the results that I showed you here. So please don't hesitate and contact me in case you have any questions or ideas for collaborations. I'm always very open to that. Thank you very much for your attention. And with that, I'm at the end of my talk. Thank you.
Thank you, Edith. It was indeed a very interesting, I would say, technology, and thanks for keeping on time and really clearly explaining all the results. So we could have some questions. I do not see any questions yet on the chat, but if you have questions, just I see. Do I see a question? So there is a question. We would prefer that you type in the question and I just ask the question, just for simplicity and managing the time. I see there is a question from Nelson. Yeah. Edith, the question is, how do we do you have any testing you do for checking durability of these materials?
Yeah, that's a broad question. Durability, maybe once you apply it in the medicine, then you would have to really do testing by implanting them into a mouse, for example, and then seeing after a while how the mechanical performance still is, whether it holds. For example, for hernia repair measures, you would implant and then see how that goes. We are just at the beginning. So at the moment, we're still doing just fundamental research. But we hope that after 4 years, we can then also move to animal testing, for example, by implanting. What we can do at the moment is just testing at room temperature in the lab how the fibers evolve over time. But this is very new type of technology, especially now in the medical field where we are applying these fibers.
Edith, would you like to read the question and answer yourself?
I can quickly see what was written there. Okay. So this about the durability. Then the next question is the regulatory concern adding toxin insecticidal compounds to the material. Yes, of course. I mean, this is something the companies that we are working with, they have to then clarify which toxins they can actually use and which not. So this is part of the industrial partners. They have to do that. So we're working with 2 big companies on this project. And yes, so regulatory, yes.
But Edith, is it not that a lot of these toxins are already known and they are approved for use in net applications?
Yes, but we are not using the pyrethroid-based toxins. We are going and using now new types,
Okay.
because that is the goal. We don't want to use standard toxins that are currently applied because these mosquitoes are resistant against. So we kind of open up now the spectrum. Thanks for sharing your research. Besides PCL, have you tried polymers with a higher melting point? Yes, we're doing that currently. So we're also testing polypropylene, PLA, and so on. And we're testing whether the drugs withstand the melt spinning of these liquid core fibers. I can unfortunately not yet share too much about which drugs withstand and which don't. But this is something we're doing right now at the moment. Then the diffusion of liquid depends on the pore size of sheaths. Have you looked into this? Thanks. Yes, we have looked into the semicrystalline structure of the polymer, especially now for PCL. Yes, so for the other polymers, we also still have to do that. So we're looking with X-rays at the size of the crystals, the orientation of the crystals, and how they affect the diffusion rate of the pores. But there are so many parameters that play into that, into the diffusion rate, also the hydrophobicity and so on, and also the chemical structure of the drug as well as of the polymer, this interaction. So that it's actually quite a complex problem. But we are studying that, yes. Then last question is, great talk. I'm particularly interested in the encapsulation of dilatant fluids. Could you explain how a fiber filled with dilatant fluid compares to encapsulating the same fluid between 2 layers of fabric in absorbing impacts? Oh, that's a very difficult question. I mean, first of all, it's very challenging to incorporate dilatant fluids because you can imagine we are actually applying quite a high pressure, and if the fluid is thickening during the application of a high pressure, that can clog the spinneret die and so on. So it needs a lot of expertise to be able to produce such filaments at the first. To answer the first thing. So it's very challenging to do that. And then once you have you can make a textile out of it. And there it depends how much of the dilatant fluid you have incorporated in that textile fabric in order to compare it with what you are mentioning, that you would have the fluid between 2 layers of fabric. I think the advantage is not that you might I think you probably overall have less effect in absorbing the impacts, but you have it all encapsulated in a fabric compared to when you just have two layers and a fluid in between. You might have a lot of leakages occurring because you have to seal the two layers. Whereas when you have the fibers really already encapsulating the fluid, then this is an advantage. At least I see it this way. I hope this answers the question.
So I think there are no more questions, but maybe I can ask one quick question. Edith, did you look into the size of active ingredient, which, or there is a limit of the size of active ingredients that can diffuse through a polymer in your applications?
Yeah, I mean, in semi-crystalline polymers, the crystals they usually range are usually in the nanometer regime, so they have a spacing maybe 2 to 5 nanometers if you have like a 50% crystallinity, which is often the case. So usually drugs that, for example, I showed you here somewhere in the one slide. I have this here. You see the radii. So this one has a radius of duration of about 5 angstroms, ibuprofen of about 3 angstroms. So we talk about sizes of drugs which are in maybe the 1 nanometer regime or below. This works very well, so they diffuse through quite easily. But then it always depends also on the polymer sheath that you're using, yeah, because the crystals they block the passage of the drugs, whereas the amorphous part in between, that's where the drugs can diffuse. So it depends a lot on the crystallinity, the orientation of the crystals, and the spacing between the crystals.
Thank you, Edith. I believe there are no more questions, and let's thank our speaker again and move to the next presentation. And if you have any burning questions, feel free to send email to Edith. Hopefully she will have some time to reply to you. I'm not sure, Shania, did we discuss about how, I mean, if the speakers are going to share the presentations or they need to just individually contact the speaker?
Yeah, we will share a recording of the presentation to all the registrants, so they'll have it accessed that way, and then they can reach out to the speakers as well.
Okay, thank you very much. So our next speaker, Dr. Liu, he is a professor in University of Manitoba. I know him for a long time. In fact, we did our PhD together in UC Davis long time back. And I really thank Professor Liu to stay awake. He's in China at this moment. It's almost past midnight. I'm not sure if I know the exact time, but it's almost past midnight. And he is speaking from there. And Professor Liu actually, since his PhD has been working on surface modification of fibers for biomedical application. He has continued working on this topic since he graduated from UC Davis. Today he's going to show us what innovative substrates he has been developing for tissue engineering and many other applications. And what is very interesting about Professor Liu's work is that not only he is publishing high quality research, but actually they are commercializing some of the technologies, which is quite important for real life application. So without any delay, I invite Professor Liu for the presentation.
Thank you, Sabir, for the kind introduction. Can you hear me okay? Okay, that's wonderful. Good morning to colleagues in North America, good afternoon to colleagues in Switzerland, and good evening to colleagues in Asia. And it's a privilege to follow Dr. Edith Perret's captivating exploration of liquid filled fibers. That's really a revolutionary bio-inspired platform that redefines the boundaries of functional textiles. So complementing Dr. Edith's nature-inspired fundamental research, I'd like to share how clinician input has shaped our basically translational research. So let me make sure I can control my slides well. So just I want to start with a question, right? So who drove the initial development of biomaterials? Chemical engineers, polymer scientists, or surgeons? So the answer is actually surgeons. It was surgeons who pioneered the first generation of biomaterials in the 1940s, repurposing readily available materials to replace diseased tissues, such as polyester for vascular grafts. So they're basically made of the same material, polyester PET, we used to make pants. And so this historical perspective underscores the critical role of clinical needs in material innovation. So I have had the privilege to collaborate closely with Dr. Loxley, a globally recognized leader in surgical innovation, whose clinical expertise and visionary insights have been instrumental in guiding our translational research. So today I will present two translational research projects that stem from his identification of clinical needs. So start with the first one. So one significant challenge he emphasized is urgent need for sensitive, user-friendly basically biosensor to monitor wound infection in situ. So this need is particularly pressing because infections not only delay wound healing, but also pose life-threatening risk if left unaddressed. So current wound dressings like silver-coated antimicrobial dressings, they often release biocides continuously, even without active infection, risking tissue damage and antimicrobial resistance. So significant progress has been made through the development of responsive systems that provide a visible response to biomedical and or physical biomarkers associated with wound infections, such as pH change and temperature fluctuation and even bacterial toxins. So Dr. Toby Jenkins of the University of Bath pioneered wound dressing for in situ detection of bacterial infection, inspired by how bacterial toxins damage human cell membranes. They designed clever mimics, lipid vesicles with phospholipid bilayer, similar to the human cells. These vesicles are loaded with the fluorescent dye, and the idea is that bacterial toxin called the cytolysine, they can break them open, releasing the dye and signaling infection. So they incorporated those vesicles into hydrogel dressing and successfully detected the bacterial biofilms. However, there's a challenge. The detection limit is for their technology is really high, around 1.0 multiplied by 10 to the power of 8 CFU per square centimeter, which is 3 orders of magnitude higher than the threshold for clinically relevant infection. So which is generally around 5 multiplied by 10 to the power of 5 CFU per square centimeter. So in other words, by the time the dressing changes color, it might already be too late for optimum treatment. Furthermore, there might be a delay in noticing the color change and seeking medical intervention. So therefore, we explore strategies for fabricating more sensitive biosensors and combining in situ diagnosis with on-demand drug delivery. To achieve this, we adopted basically a more sensitive strategy, right? So we want to target the specific bacterial enzymes known for their remarkable catalytical ability at body temperature. So we designed and synthesized the new chromogenic probe based on hemicellin This probe, we call it HCY dye, features an ester group which can be hydrolyzed by bacterial lipase, facilitating a visible color change from yellow to red in the presence of lipase-generating bacteria. So specifically in this case, we expose the dye to lipase at the body temperature, 37 degrees C, and the HCY dye is cleaved, right? And this cleavage leads to increased intermolecular charge transfer between the phenolate ion with the semi-silane group, accompanied by a characteristic new absorbance at around 575 nanometer, which basically is accompanied by a visible color change from yellow to red. So the higher the concentration of lipase, the darker the red color. Similarly, we designed a prodrug based on ciprofloxacin. This carboxylic acid group of ciprofloxacin was end-capped with 6-azidohexanol, right, to form an ester. So initially, during the structural optimization, we evaluated multiple complex modification strategies, including triglyceride derivatives. So yet, interestingly, experimental data shows that the simpler ester linkage exhibited higher prodrug activation efficacy. So then we stick to the simpler ester functional group instead of more complicated triglyceride structure. So to enhance the sensitivity towards bacteria, we use the electrospun nanofibers with a high specific surface area as a matrix. So we incorporated the chromogenic probe and the prodrug into the polyurethane solution and the electrospun the solution into nanofibers. So that video shows electrospinning in action. So as revealed by SEM micrographs, the nanofibers in the membrane are in nanometer range with an average diameter of 480 nanometer. So let's see how the theranostic membrane performs in action. So we conducted the in vivo study mimicking the wound environment. After incubating the membrane with the Pseudomonas aeruginosa, a pathogenic bacterium known to produce lipase, for 4 hours at body temperature, we observed the visible a color change from orange to green. Interestingly, the same membrane exposed to E. coli top 10, a strain that doesn't generate lipase, we consider that strain as non-pathogenic. So the membrane remained its original color. We also evaluated the antimicrobial activity. The membrane effectively eliminated Pseudomonas 100% at the 4-hour time mark. But in contrast, minimal impact was observed on E. coli top 10, as indicated by the black column. So these findings strongly suggest that the membrane is selectively activated by lipase-secreting bacteria. So this selective activation facilitates an on-demand release of the drug and the color change of the chromogenic probe at the infection site. So as demonstrated by the WST assay, both the theranostic membrane, number 3 and number 2, the polyurethane membrane, appear to be non-cytotoxic to fibroblasts, the skin cell. So for the first time, this research presents a nanofibrous theranostic antimicrobial dressing. It functions as a one-stop shop for wound care by offering both diagnosis and treatment in a single dressing. So we did decrease the detection limit, as I mentioned initially, as a gap for the research, right? So we decreased the detection limit to 10 to the power of 7 CFU per square centimeter, but it's not quite there yet. So we adopted 3 more strategies to further boost the detection sensitivity. The first one is to similar to what Edith mentioned, like a core shell structure. So we enriched the chromogenic probe in the shell of core shell structured nanofiber. And then another is to add a polymeric dopant, polyvinyl pyrrolidone, PVP, in the shell as a dopant to enhance the charge transfer in the HCY dye, enabling faster and more sensitive detection of bacteria. As shown on this slide, the detection limit for single SPU, single spun polyurethane, the detection limit is 5 micrograms per ml for model lipase. Whereas the core shell structure nanofiber with or without PVP could detect lipase at a concentration as low as 1.25 micrograms per ml. So after 8 hours of incubation. So that's a 4-fold increase in sensitivity. And upon exposure to bacteria, the PVP containing membrane showed a darker and more vivid, as you can see here. PVP containing membrane shows darker and more vivid color change compared to the single electrospun PU and the core shells PU. Meaning that PVP also enhanced the discernibility of the color change from yellow to green, making it easier to detect the bacterial presence. So we did basically adopt the last strategy to finally reach a clinically relevant threshold of infection, which is around 5 multiplied by 10 to the power 4 CFU per square centimeter, by adding a surfactant, because lipase is known to catalyze ester cleavage at the oil-water interface. So the patent has been filed for this technology, and a startup company, Perret Wound Care, from state US, licensed the technology in 2018 and has been working collaboratively with us to transform this research to a clinical product, which is nearing commercialization. So we also worked with the largest company in the space of artificial skin, Integral Life Science, to provide solutions for wound infection problems. So Perret is commercializing technology as WoundQ, which is currently FDA registered. The company has 2 pre-market purchase orders to sell into the US Veteran Affairs System and the pediatric hospitals. Perret is now gearing up for clinical trials set to begin in January 2026, with a full market entry planned after those trials conclude in the spring. So we also got feedback from Perret that in some clinical scenarios, differentiating between gram-positive and gram-negative bacteria is crucial for selecting the appropriate antibiotic therapy. So I want to showcase our recent progress along the line. We explored the fascinating properties of polydiacetylene, PDA, to achieve that goal. So PDA chain possess a unique arrangement of alternating triple bond and double bond. So within their backbone, this configuration creates a system of conjugated electrons. These chains are further adorned with the customizable side group that significantly influence the perceived color. So here comes the key. When those side groups bind with some specific target, right, so they will become bulkier and disrupt the conjugation, right? And then the conjugation is shortened and the color will be changed. So we have successfully harnessed this property to differentiate between gram-positive and gram-negative bacteria, including pinpointing a specific strain of Pseudomonas. So here comes a little bit of procedure. We first blended a diacetylene monomer with a medical grade polyurethane in DMF and the THF solvent, and then electrospun the solution into nanofibers. So finally, UV irradiation was used to trigger the polymerization of the monomer, forming the PDA, which was then modified with the antibacterial peptide to target gram-negative bacteria. So here comes the result. The nanofibrous membrane turned red after 2 hours of incubation, with the gram-negative bacteria, including E. coli and the Pseudomonas, but it remained as blue or purple with the gram-positive bacterium as aureus. So furthermore, we could pinpoint a single ATCC strain within Pseudomonas by conjugating onto the PDA polymer with the two aptamers targeting Pseudomonas aeruginosa. So as we can see from the results after conjugating two aptamers, so the membrane can really pinpoint a specific strain within Pseudomonas species. So that really just like highlight the strain level specificity, right? So which can enable precise anti-infection therapy. So this work has been published as an invited contribution to in advanced functional materials. So to wrap up, we developed the smart Theranostic wound dressing. It detects bacterial infection at a clinically relevant level with high sensitivity. So using PDA color changing properties, we can identify specific strains within a species, like Pseudomonas aeruginosa ATCC 27853. So here comes the second project I want to share. So while the Theranostic wound dressing demonstrates efficacy in infection control, but what if we want to achieve complete tissue regeneration? So for that purpose, we need something like a little bit beyond passive protection. Demands a scaffold that actively promotes cellular repair and functional restoration. So a little bit of background. Severe burns destroy the skin's architecture, leaving patients vulnerable to infections, scarring, and even life-threatening complications. So traditional treatments such as autologous skin grafts remain the gold standard, but they are limited by donor site availability. So our vision is ambitious. We want to create a scaffold that's immune protected, rapidly vascularized, and truly off the shelf, ready to use in emergencies. But more importantly, we want to move beyond wound closure, not just repair, but towards scarless regeneration, restoring the full structure and the function of native skin. So now let's delve into the specifics of our new skin substitute. Imagine a two-layered scaffold meticulously crafted by synergistic use of nanofiber electrospinning and 3D bioprinting to create an optimized microenvironment for cell attachment, proliferation, and wound regeneration. So, but the magic lies in the cells that would populate the scaffold. So we are engineering human dermal skin cells to be non-immunogenic. That means these cells won't trigger rejection by the recipient's body, and also they are self-limited. They're programmed to be dependent on specific amino acids, which they can't produce. So then, therefore, we can really induce their apoptosis or cell death after a certain time. So beauty lies in the design of the nanofibers, right? So we, according to study, fibroblasts thrive on larger microstructures, while keratinocytes prefer finer nanofibers. So leveraging this insight, we propose a dual layered structure, right? A nanofibrous membrane, right, for the epidermis, and 3D bioprinted collagen or gelatin scaffold for the dermis. So by precisely tailoring the diameter of these scaffolds, we can create a microenvironment optimized for each cell type. So to further enhance functionality, we plan to encapsulate growth factors and the life-limiting amino acids in the shell or core of the core shell structure nanofibers. So this approach can help us achieve precise engineering of the gradual replacement of the anogenic skin cells with autologous counterparts, mimicking natural skin regeneration. So our early results are promising. So in the domain of cell genome editing, we initially employed a kidney cell like a HEC 293T as a model system to establish and validate our experimental approach. So we use like a T cell suppression assay to evaluate the effect. So we basically co-culture T cell with the engineered model and the wild type HEC cell. So the results show that the proliferation of T cell drops from 41% to just 3.7% in the case of engineered HEC cell. So confirming strong immunomodulatory capacity of the engineered cells. So here actually we are making a stride in engineering skin cells using the validated strategy. So on the scaffold end of research, we are now moving towards more complicated constructs. RTPEG interfaces and the hierarchical dermal layers, including the vascular channels for neovascularization of the skin substitute. So for the epidermis, traditionally made like electrospun nanofibers, they are very dense and compact. It's not conducive for cell infiltration. So therefore, we expanded them using a gas forming technology, using basically sodium borohydride, and inducing its hydrolysis in acidic condition. So we release hydrogen gas, creating larger pores. So as you can see from the SEM micrographs, the expanded membranes exhibit a significantly higher porosity as 70% versus 25%, and a larger pore size. Additionally, the interlayer spacing increased dramatically from 5 micron to 196 micron, indicating a substantially enhanced three-dimensional architectural complexity. So this allows keratinocyte to attach and spread with much higher cell density, as you can tell here. So the cells marked by the yellow color are much happier on the surface of expanded membrane. So we conducted a migration assay to evaluate the cell migration across two distinct membrane configurations. So here comes a little bit the methodology. So primary keratinocyte cells were first labeled with a cell trace dye, and subsequently those cells were seeded into two chambers separated by a gap, which is around 0.5 millimeter. So, and the culture overnight on tissue culture coated well surface. So a second day, we remove the insert, right, and then we put the membrane, be it flat membrane or expanded membrane, into the gap. So with the XY plane oriented parallel to the surface of culturing flask. So following overnight culturing, so cells in each chamber form basically monolayer culture. And so we want to look at the result. The fluorescent microscope image was captured on day 0, basically time 0, and time 7 after a week. Right, so those red colors basically means the presence of cells. So under the flat membrane condition, right, so we can tell the cells basically are saturated. They try to close the gap, right, migrate towards the membrane, but quickly upon saturation of flat membrane, so they kind of felt the resistance. They started to retract, right, and they actually move towards the opposite direction, right? Instead of going towards the center, they actually retracted away from the center, right? And as you can tell from the fluorescent image. So with the left side exhibiting a more dominant effect of that retraction. So whereas in the case of expanded membrane, so because it has multiple layers for the cell to migrate into, so a zoom in the view showing that labeled cells moved towards the center of the gap in the membrane. And we use Z stack imaging to show the multi-layer presence of the cell. Right, so this video shows a layer from layer number 1 from the bottom to 30 layers above, and which with the interlayer gap of 20 micron, as you can tell, cells present on different focal lengths. Right, so they are actually migrating from the bottom to the top. So we also extended the incubation to 2 weeks. Then cells were fixed and stand with the DAPI dye, which stands the cell nuclei, blue. So the DAPI stand nuclei blue color. So the red signal corresponds to cell initially labeled with cell trace dye, and we can tell from for the case of flat membrane, the majority of the cells actually stuck at the edge, whereas in the case of expanded membrane, they migrated into center, and because the color will be lost after beyond 5 generations of division, so we start to see the fading of the color red, and the presence of the blue color, independent of the red color, means we have more daughter cells beyond the 5th generation. So that indicates a stronger or more active proliferation of the cells in the presence of the expanded membrane. So as you can tell from the side view, right? So the blue color really is pretty intense, right? And they made the whole layer, right? And even single cells are not very visible. So that indicates the proliferation of migrated cells was much higher in the expanded sample after 2 weeks. Oh, sorry, move us too fast. So for dermal matrix fabrication, this goes a little bit beyond that nanofibrous membrane. So I will probably stick to the nanofibrous membrane. So this animation illustrates theoretical spatial distribution of keratinocyte across both membrane systems. So the expanded membrane demonstrated enhanced suitability for supporting keratinocyte migration and proliferation, right? Because they have more layers to migrate into, and the pore size are bigger to allow the membrane to penetrate into the membrane. So it's much more conducive than the flat membrane. So for the cell 3D bioprinting part, so probably I will skip because it's not very related to the fibers. But the idea is we can laminate the nanofibrous membrane with the 3D bioprinted dermal scaffold. So we can obtain a novel engineered cell-laden skin substitute. So the wound regeneration with this skin substitute would go as follows. Engineered cells will first basically populate the wound, initiating the regeneration. So after 2 to 3 weeks, these cells will die, right, due to the amino acid depletion. Right? As I mentioned, we engineer them to be dependent on that amino acid encapsulated in the nanofiber or 3D printed scaffold. So this will create a space for the autologous cell, like a patient's own skin cells, to take over and complete healing, minimizing scarring. So we will also use adipose-derived stem cell to generate all the skin appendages, including hair follicles, sweat glands, and possibly the sebaceous glands. So together, these innovations demonstrate how fiber-based platform can be rationally designed to support both therapeutic function and tissue regeneration, paving the way for next generation wound care technologies that are both intelligent and biologically integrative. So with that, so I'd like to express my sincere gratitude to my talented graduate students. Obviously, those innovations won't be possible without the hard work and dedications of all my students. So a big shout out to my fantastic collaborators. And obviously, I need to also a huge thank you to all funding support. Last but not least, thank you for your focused attention. So I now welcome any questions or insights you may have. Thank you very much.
So thanks, Song. It was very interesting. All the smart wound dressings you have been developing, started with the colorimetric identification and then finally the smart regenerative tech ideas you have. Questions from the audience? Maybe I could start the questions. In the first presentations you showed, when you have these lipases are released from the bacteria, which then trigger the color response. Do you also think about releasing drugs as the color changes?
Yeah, so that's a great question. So can you hear me okay?
Yeah.
Yeah, so basically, besides that chromogenic probe, we designed a probe drug based on antibiotic, which is called ciprofloxacin. Right. So let me go back to that part.
So maybe I missed that point, but somehow they should be coupled together. The color change with the drug release is something which is.
Yeah, so this is a color, it's a dye, right? So and then we actually also designed a prodrug, right, which is capped by 6-azidohexanol, right, from an ester group, like and capped that carboxylic acid group of the active antibiotic.
Okay.
Right. Yeah. You know what? Initially, we, as you can probably see, that we have azide handle here.
Yeah.
Initially, I wanna covalently click them onto the textile substrate. So, however, we found out that the strategy that didn't work because the concentration of the clicked molecules are too low, right? So, and wasn't sensitive enough. So, end up we decided to just physically blend into nanofibrous membrane, but we blend them into the core shell structured nanofibers, so we can basically tune both the sensitivity and the speed of the release.
Thanks, Song. Do we have any questions from the audience? Feel free to type in the questions. Maybe I continue with one more of my questions so that the audience can warm up a little bit. You mentioned that the first idea is almost towards commercialization, huh? So
yes.
what are the hurdles at this moment which the industry has to face before it can commercialize the technology?
Yeah, that's a great question. Yeah, so I think that's really, you know what, I also had some other technologies which was patented, and so, but for this specific like a drug, like a theranostic membrane, so it started from the input of clinician, right? So it's like a clinical needs driven. So the acceptance of the market, like penetration, obviously now we don't have the penetration yet, but the hurdle really lies in the registration, FDA registration. So, but now the product is self-registered with FDA, but still they need to complete the clinical study, right? Collect enough data. But the acceptance of the technology by doctors, users, right, and the patient is really good, right? So as I mentioned, actually Perret got 2 pre-market purchase orders totaling around $1 million, right? So really means even before the product is available for sale for like a market entry, so they already got purchase orders, right? And really just speaks to the need, right? Because it's very much needed, right? So and then it works. And another thing, a hurdle, as you mentioned, is really just third-party validation. So because our lab validated the technology, but we need a third-party validation. Fortunately, it was done last year by a European company. So far,
Okay.
I may not be able to disclose the name of the company, but they were very interested. And then we sent them the product, and they validated the product using their own testing methods, and found out it really just has a very good like a specificity in detecting infection without like a false positive. Yeah, I think to your question, really just a hurdle is um, how the technology would work in real like a case scenario, right? So in a real wound, right? So so that has been tested, but it's still in labs, right? So they are moving towards clinical study beginning January 2026.
Would you briefly explain what is the FDA approval process like, so that the audience can have an idea how long it takes from invention all the way to commercialization through FDA approval? Very
Well,
briefly.
yeah, that's definitely an interesting and a great another great question. So here is really a good strategy I think Perret has adopted, because for medical device, there are many classes, right? Class 1, class 2, class 3, right? So the higher the class, and the more demanding and the time consuming to get the product registered. Um, but their strategy is they first I guess register the product as a class 1, right? And then it will move much faster, right? And in the meantime, they can start to like sell the product as a class 1, like a biomedical device, right? And then in that process, they can collect data, right? To say, okay, whether or not detect the infection, shows color change. So in other words, the product is currently self-registered as a class 1 product. They didn't claim it should detect infection. Okay, so they didn't claim that. But I, as a researcher, I can present what we have done, but I won't speak on behalf of the company. Right? So I only speak on behalf of myself. Right? So what we have done in our lab, what data we have collected, but the company doesn't claim anything. Okay, for their like a color detection, like a color change or infection detection. Right? So, but they just claim it as like a common wound dressing, right? And until they collect data, right, to clinical data shows it does like a change color and the sufficient data to satisfy FDA requirement for class 2 medical device, and then they can make the claim. But this procedure basically will be much longer, right? So their strategy will greatly streamline the process.
Thanks, Song. I see there are at least one question and a comment, so maybe you can go ahead and read the question and comment.
Okay, maybe I need to stop sharing, and so I can read the comment. Okay, so thank you. So what are concrete application illnesses where visualizing bacterial infection with fluorescent dye is of interest? Wound dressing are often changed regularly. Yeah, so that's a great question, right? So from the clinical perspective, I have that like a surgeon friend, Dr. Salvesh Laksani, as I highlighted early in my talk. So according to him, so in a lot of antimicrobial dressings, they claim they can be placed in they can be put in place for 7 days. But doctor would tell you won't happen, right? So generally 2 to 3 days, or some cases they change on daily basis based on types of the wound. Some highly exudating wound, like they have a high amount of exudates, you have to change every day. Right? But for a little bit low, in like a medium exudating wounds, you can keep the dressing on and for 2 to 3 days. For example, I mentioned the Acticoat, right? So the silver containing antimicrobial dressing. So even though they claim that you can like allow them be in place for 7 days, but surgeons don't generally do it, right? So they basically change the dressing 3 days, but that gives them peace of mind, right? So they don't because the antimicrobial dressing can like make sure the wound environment is sterile, right, and doesn't have any infection. So for like as I mentioned, for pediatric hospitals and for veteran affairs system, right, and the diabetic wounds, right, and for off patient, right, so they're not hospitalized, right, so they don't change the dressing as often, like every day or 2 to 3 days, because it's labor intensive, right? So nurses will be very busy in changing dressing, and the dressing change process is very painful, right? And so I think to really just answer your question, I would give you an example of second degree or even third degree burns, right? And especially with a large area of burns, right? So that's related to Savage, like Sabir's work for flame retardant, right? So firefighters, right? So when they experience such a like a large area or high level degree of burns, right? And those burns pretty hard to heal, and you don't want to change dressing every day or every 3 days. You want to keep the dressing longer to allow the new tissue to mature. So otherwise, the changing process gonna cause a secondary trauma, right, and cause those newly formed tissue to be damaged again.
Thanks, Song. Maybe a last comment and a question from Professor Gang Song.
Yeah, thank you, Dr. Sun. I'm happy I saw you jumping in during my talk, and really I was very happy to see you here. Thank you for your great comment. And is it possible to have electrical signals? Yeah, I think that's a wonderful question, and because a lot of studies show electrical signal will trigger cells to migrate, right? Besides the chemotaxis, right? And we have electrical current which can stimulate cells to migrate and proliferate, right? So I think it's possible, right? And some studies show that if they use like inorganic salt or metals, so they can create some localized electrical current, like based on redox reaction, right? And that can stimulate cell growth and the proliferation. And so, yeah, I think if we use some conductive wires made of like fibers, right, and we can really just move towards that direction.
Have we got any final questions for both speakers?
Oh, Dr. Song, you just
yeah, I
returned
think
to
he
US.
just like here. Yes, I just see that he may have gone back home.
Okay.
So I see no more questions from the audience, and I also have no more questions. I really thank both speakers, especially Song, for staying awake. It's almost 1 o'clock in the morning. I would say early morning. So yeah, I thank both the speakers for their wonderful presentations. If the audience have questions, they can just send email to the speakers. And again, thanks all the audience to stay behind and listen to the presentation. And yeah, so I look forward to the next meeting, which is planned for early next year. So again, thanks again all, and have a nice morning, evening, and night for all of you. Thank you. Bye.
Sounds good.
Bye-bye.
Thank you. Okay. Bye.